Welcome to the deep dive. Today we're getting into a system that works constantly, every single minute, to keep you alive. Your respiratory system.
Hmm.
And it's so much more than just breathing. It's this incredibly intricate dance of physics, chemistry, and biology happening inside you right now.
It really is.
Our mission for this deep dive is to really understand the core mechanics of how you breathe specifically, how your body takes in oxygen and gets rid of carbon dioxide. We're using Chapter 13, Respiratory Physiology, from Vander's Human Physiology, as our guide.
Right. The goal is to give you a clear, solid understanding, almost like a top-tier physiology lecture, but, without needing any slides or diagrams. We'll stick to analogies only when the textbook would.
And it's important right off the bat to clarify what we mean by respiration today.
Absolutely. There are two main meanings. There's internal or cellular respiration. That's how your individual cells use oxygen for energy. That's not our focus today. We're diving into pulmonary physiology. That's the exchange of oxygen and carbon dioxide between your whole body and the outside air. Basically, the journey of every breath you take.
And what's so amazing is how this system works hand in hand with your circulatory system. They're like a team, delivering oxygen and nutrients and hauling away waste products.
Exactly. And you'll see how basic physical laws, things like pressure and flow, govern everything from how gas is attached to your blood cells to how your lungs actually inflate. Okay, so let's start by unpacking how this whole system is put together.
Sounds good. So we have the lungs themselves, two of them, right and left, divided into lobes.
But the real workhorses, the main functional units, are the alveoli. Picture about 300 million incredibly tiny air-filled sacs deep inside your lungs. That's where the crucial gas exchange with your blood happens.
300 million, that's hard to even imagine. And the simple act of moving air in and out is breathing. So inspiration is breathing in, getting air to the alveoli.
Right, inhalation. And expiration or exhalation is breathing out, moving air away from the alveoli. One breath in and one breath out makes up one respiratory cycle.
Okay.
And just to give you a sense of scale, when you're resting, you move about 4 liters of air in and out of those alveoli every minute. At the same time, your heart is pumping around 5 liters of blood, your cardiac output, through the capillaries surrounding those alveoli.
Wow. Pretty closely matched.
Very. But get this. During heavy exercise, that airflow can jump up 20 times, and blood flow can increase 5 or 6 times. The system is incredibly adaptable.
So let's trace the path air takes. It starts at the nose or mouth.
Then goes through the pharynx. It's the area at the back of your throat, shared by air and food.
Got it.
From there, it enters the larynx, or voice box, which houses your vocal cords. Then down the trachea, the windpipe, which is a fairly long tube.
Okay.
The trachea then splits into 2 main bronchi, one heading into each lung.
And inside the lungs, it keeps branching.
Oh yes, over 20 generations of branching, getting narrower and narrower. The trachea and the first few branches, the bronchi, have rings of cartilage to keep them open, like rigid pipes.
Makes sense.
But eventually, the airways lose that cartilage and become bronchioles. These are surrounded by smooth muscle, which means their diameter can change, kind of like arterioles in the circulatory system.
Ah, okay. Control points.
Exactly. These bronchioles lead to terminal bronchioles and then to respiratory bronchioles, which is where you first start seeing alveoli budding off. Then come alveolar ducts, with more and more alveoli, ending in these clusters that look like tiny bunches of grapes called alveolar sacs, made entirely of alveoli.
So we can think of the airways in 2 main zones.
That's a great way to put it. First, the conducting zone. That's everything from the trachea down to the terminal bronchioles. Think of it as the plumbing. No gas exchange happens here. Its job is to provide a clear, low-resistance path for air. Plus it warms, humidifies, and cleans the air. Your vocal cords are here too, for sound.
And the cleaning part.
Right, it has defenses. Hairs in your nose, mucus lining the airways, and tiny hair-like cilia that constantly beat upwards, pushing mucus and trapped particles towards your throat to be swallowed. It's called the mucus escalator. Smoking, by the way, really damages these cilia.
Ah, hence the smoker's cough.
Precisely. And then there are macrophages and immune cells patrolling for anything that gets past. Plus, if irritants get deep, the bronchioles can actually constrict to try and block them from reaching the alveoli.
Smart design. And the second zone.
That's the respiratory zone. It starts where the alveoli first appear, the respiratory bronchioles, and includes the alveolar ducts and sacs. This is purely for gas exchange.
Now, you mentioned problems like cystic fibrosis earlier.
Yes, the cystic fibrosis, or CF, it's a genetic disorder affecting a protein, the CFTR protein, which is a chloride channel. This messes up the watery fluid layer beneath the mucus. So the mucus becomes really thick and dehydrated, clogging airways and leading to infections. It's a serious challenge for the conducting zone's protective function.
Understood. Let's zoom in on those alveoli then, the gas exchange hubs.
Okay. They're incredibly thin-walled sacs. The wall itself is mostly made of type I alveolar cells. These are super flat epithelial cells, just one cell thick, perfect for diffusion.
And you mentioned another type.
Yes, scattered among them are type 2 alveolar cells. These are crucial because they produce a surfactant. Think of surfactant as a detergent-like substance.
Okay.
We'll come back to why it's so important.
Okay.
The outer wall of the alveoli is covered in capillaries, which is a tiny, tiny space between the alveolar cell and the capillary wall. The total barrier for gas exchange air to blood is unbelievably thin, about 0.2 micrometers. That's much, much thinner than a red blood cell diameter.
Wow. And the surface area.
If you laid all your alveoli flat, it would cover a tennis court. This combination, incredibly thin barrier, enormous surface area, is what allows for such rapid and efficient gas exchange.
Amazing. Now, how do the lungs actually sit in the chest?
Right, the lungs are housed in the thorax, or chest cavity. It's a closed compartment, sealed off from the abdomen below by the diaphragm, a large dome-shaped muscle. The walls are the ribs, spine, sternum, and the intercostal muscles between the ribs.
And each lung has its own covering.
Exactly. Each lung is enclosed in a separate double-layered sac called the pleura. The layer stuck to the lung is the visceral pleura. The layer lining the inside of the chest wall and diaphragm is the parietal pleura.
Okay, 2 layers. Is there space between them?
Just a potential space, containing a very thin film of intrapleural fluid, only a few milliliters. It acts like lubrication, letting the lungs slide smoothly against the chest wall as you breathe.
You mentioned an analogy.
Yeah, think of pushing your fist, the lung, into a fluid-filled balloon, the pleural sac. The balloon surfaces, pleura, are very close, separated only by that thin fluid layer. Or like 2 wet microscope slides, they slide easily, but resist being pulled apart.
So that fluid creates a sort of suction.
Precisely. It creates a negative pressure in that intrapleural space relative to the atmosphere. This intrapleural pressure, PIP, is typically around -4 Chi between breaths. It's this negative pressure that effectively links the lungs to the chest wall, making them move together. It's absolutely crucial.
Which leads us neatly into ventilation and lung mechanics.
Right. Ventilation is just the mechanical process of moving air between the atmosphere and the alveoli. And air moves by bulk flow, always from higher pressure to lower pressure.
Like water flowing downhill.
Exactly. The equation is simple. Flow, F, equals the pressure difference divided by resistance, R. Same idea as blood flow.
So for air, the pressure difference is between
the pressure inside the alveoli, PALV, and the pressure of the atmosphere outside, SPAT. So flow, PALV, PATM, R.
And we treat atmospheric pressure as 0 for simplicity.
Yes, all respiratory pressures are usually given relative to atmospheric pressure, which is about 760 millimole-G at sea level. So PATM midland 0 millimole-G. If PALV is negative, less than PATM, air flows in. If PALV is positive, greater than PATM, air flows out. Between breaths, PALV is 0, so no air flows.
And what causes these pressure changes inside the alveoli?
This comes down to a fundamental gas law, Boyle's law. It states that for a fixed amount of gas at a constant temperature, pressure and volume are inversely related. P1V1 equal P2V2.
So if you increase the volume of the container, the pressure drops. Decrease the volume, pressure rises.
Exactly. And your lungs are the container. Changes in lung volume directly cause the changes in alveolar pressure that drive airflow. It's that simple, conceptually.
Now, you mentioned that negative intrapleural pressure holds the lungs open. Can we unpack that a bit more?
Sure. Think about the forces at rest between breaths. Your lungs are elastic like balloons, and they naturally want to recoil inwards to collapse.
Okay.
But your chest wall is also elastic, and it naturally wants to spring outwards.
So they're pulling in opposite directions.
Exactly. This opposition pulls the visceral and parietal pleura slightly apart, creating that subatmospheric negative intrapleural pressure, PIP, that -4 milli-G we mentioned.
And this negative PIP acts across the lung wall.
Yes. The pressure difference across the lung wall is called the transpulmonary pressure, PDP. It's calculated as PALV minus PIP. Since PIP is negative, PDP is positive. This positive PDP is what physically holds the lungs open and prevents them from collapsing.
Ah, I see. So PDP equals 0.4 equals +4 millimole-Hg at rest.
Precisely. And if something breaks that seal, like a puncture wound to the chest.
Air rushes into the intrapleural space.
Right. That's a pneumothorax. The intrapleural pressure equalizes with atmospheric pressure, becoming 0. Suddenly, PIP is no longer negative.
Which means the transpulmonary pressure becomes zero.
Correct. And without that positive pressure holding the lung open, its natural elastic recoil takes over and the lung collapses.
Okay, that makes sense. So let's walk through a normal breath. Inspiration first.
Inspiration is an active process. It starts with signals from your brain telling specific muscles to contract. The main one is the diaphragm.
The dome-shaped muscle at the base of the chest.
That's the one. It contracts and flattens, moving downwards.
Right.
At the same time, the external intercostal muscles between your ribs contract, pulling the ribs upwards and outwards.
So the whole chest cavity gets bigger.
Yes, the volume of the thoracic cavity increases.
Yeah.
As the chest wall expands, it pulls the parietal pleura outwards. Because of that fluid linkage, the intrapleural pressure, PIP, becomes even more negative, maybe -7 millimol Hg.
Which increases the transpulmonary pressure? PTP.
PDP becomes larger. Yog 0 -7 equals +7 millimol Hg. This increased distending pressure pulls the lungs open, making them expand passively along with the chest.
And as lung volume increases, Boyle's law.
You got it.
Yeah.
Increased lung volume means the pressure inside the alveoli, valve, drops below atmospheric pressure, say to melanoid 1 milli-LHG.
And that pressure difference makes air flow in.
Exactly. Air flows into the lungs until PALV rises back to 0 millimHg, equal to atmospheric pressure. And that's the end of inspiration.
Okay, so expiration at rest.
Usually passive. The nerve signals stop, the diaphragm and external intercostals relax.
The chest wall naturally recoils inwards.
Yes, due to its elasticity. The thoracic volume decreases. PIP becomes less negative, returning towards -4 millimHg. The transpulmonary pressure decreases.
So the lungs are less stretched open?
Correct. And because lungs are elastic, they passively recoil back to their smaller resting volume.
Boyle's law again, smaller volume.
Means higher pressure. PALV becomes positive, maybe +1 millimHg, and air flows out of the lungs until PALV equals padded millimHg, 0 millimHg again.
Simple and elegant when it's passive. What about forceful expiration?
That's active. It involves contracting the internal intercostal muscles, which pull the ribs down and in, and also contracting your abdominal muscles. This pushes the diaphragm up forcefully, rapidly shrinking the thoracic volume, generating much higher positive alveolar pressures and forcing air out quickly.
Okay, let's talk about lung compliance. What is that exactly?
Compliance is basically a measure of how stretchy or distensible your lungs are. Technically, it's the change in lung volume divided by the change in transpulmonary pressure, VL.
So high compliance means they stretch easily.
Yes. Low compliance means they're stiff, harder to inflate. Think of a stiff balloon versus a floppy one. If lungs are stiff, it takes much more muscular effort to achieve the same amount of inflation. People with low compliance diseases like fibrosis often breathe shallowly and rapidly.
What determines compliance?
Two main things. One is the amount of elastic connective tissue in the lungs. More tissue, like scar tissue in fibrosis, makes them stiffer, reducing compliance. But the major factor is the surface tension of the liquid lining the alveoli.
Surface tension, like on water.
Exactly. The alveoli are lined with a thin film of liquid, mostly water. Water molecules attract each other strongly. This creates a force, surface tension, that tries to minimize the surface area, essentially. It makes the alveoli want to shrink or collapse, and it resists stretching.
That sounds like it would make breathing harder.
It would, significantly.
Yeah.
But this is where surfactant comes in. Remember those type 2 alveolar cells? They secrete surfactant, which is a mix of lipids and proteins.
And it acts like a detergent?
Sort of. It gets between the water molecules at the air-liquid interface and reduces their attraction to each other. This lowers the surface tension dramatically.
Making the lungs more compliant, easier to inflate.
Exactly. It hugely reduces the work of breathing. Interestingly, taking a deep breath actually helps spread surfactant and stimulates its secretion. That's why sighs can feel good.
Is that why surfactant is crucial for newborns?
Yes. Respiratory distress syndrome of the newborn happens in premature infants who haven't produced enough surfactant yet. Their lungs are incredibly stiff, low compliance, and breathing is exhausting, often requiring ventilation support and administration of artificial surfactant.
Surfactant also helps keep alveoli stable, right?
Yes, based on the law of Laplace. Without surfactant, smaller alveoli would have higher inward pressure due to surface tension and tend to collapse into larger ones. Surfactant uniquely reduces surface tension more effectively at smaller volumes, equalizing the pressure between small and large alveoli and preventing collapse.
Okay, moving on to airway resistance, how much friction does air encounter?
Normally, very little. Airway resistance is usually so low that a tiny pressure difference like 1 millimHg is enough to move a normal breath's volume, about 500 millibal.
What determines resistance?
Overwhelmingly, the radius of the airways. Just like blood vessels, resistance is inversely proportional to the radius to the fourth power. Small changes in radius have a huge effect.
What normally affects the radius?
During inspiration, the increasing transpulmonary pressure actually pulls the airways open wider, reducing resistance. The elastic tissue surrounding the airways also exerts an outward pull called lateral traction, helping to keep them open as the lungs expand.
Are there other controls, like nerves or hormones?
Yes. Epinephrine, for example, relaxes airway smooth muscle, causing bronchodilation and decreasing resistance. Certain inflammatory chemicals, like leukotrienes, cause bronchoconstriction, increasing resistance.
Which is relevant for asthma.
Definitely. Asthma involves chronic airway inflammation that makes the smooth muscle hyperresponsive. Triggers can cause strong bronchoconstriction, drastically increasing resistance and making breathing difficult. Treatments often include anti-inflammatories and bronchodilators.
What about COPD?
Chronic obstructive pulmonary disease, COPD, also involves increased resistance, but usually not from smooth muscle spasm like in asthma. In emphysema, there's destruction of lung tissue, leading to collapse of the smaller airways, especially during expiration. In chronic bronchitis, there's excessive mucus production and inflammation thickening the airway walls.
Let's briefly cover lung volumes. We measure these with a spirometer.
Most of them, yes. The air moved in a normal breath is tidal volume, VOT-2 is about 500 millimol. The extra air you can inhale after a normal breath is the inspiratory reserve volume, IRV. The extra air you can exhale after a normal breath out is the expiratory reserve volume, ERV. The air always left in your lungs, even after forcing out as much as possible, is the residual volume, RV. You can't measure RV directly with simple spirometry.
And combinations of these give capacities.
Right. Functional residual capacity, FRC, is the air left after a normal exhale, VRV plus RV. Vital capacity, VC, is the maximum air you can move in one breath, IRV plus VT plus ERV. VC is important in lung function tests.
Like the FEV1 test.
Yes. Forced expiratory volume in 1 second, FEV1, measures how much of your vital capacity you can exhale in the very first second of a forced breath out. It is a measure of airflow speed.
How does this help diagnose problems?
In obstructive diseases like asthma or COPD, resistance is high, so it's hard to get air out quickly. FEV1 is significantly reduced, usually less than 80% of the VC. In restrictive diseases like fibrosis, the problem isn't airflow, but reduced lung expansion. So the VC itself is reduced, but the FEV1-VC ratio is often normal or even high because they can get the small volume out quickly.
Okay, that distinction is clear. Now, let's think about how much fresh air actually gets to the alveoli for gas exchange.
Right, that's alveolar ventilation, VA. It's not the same as your total ventilation per minute. Your minute ventilation, VE, is just tidal volume times respiratory rate. Example, 500 ml breath by 12 breathsmen equals 6,000 millimin.
But not all of that 500 milliway reaches the alveoli.
Correct. Some of it just fills the conducting airways, the trachea, bronchioles, where no gas exchange occurs. This volume is called the anatomical dead space, VD, and it's roughly 150 milliliters in an average adult.
So that 150 millivale is essentially wasted with each breath in terms of gas exchange.
Pretty much. There can also be alveolar dead space if some alveoli are ventilated but don't have blood flow, but that's small and healthy lungs. The total physiological dead space is mostly just the anatomical dead space.
So the useful ventilation, alveolar ventilation, is less than minute ventilation.
Yes. VA, tidal volume dead space, X respiratory rate. Using our example, 500 millil, 150 millil by 12 breathsmen equals 4,200 millilane.
That's significantly less than the 6,000 milliliter minute ventilation.
It is. And this highlights something important. Increasing your depth of breathing, tidal volume, is much more effective at boosting alveolar ventilation than just increasing your breathing rate.
Why is that?
Because with each breath, you have to move that fixed 150 milliliters of dead space air first. If you take bigger breaths, the dead space volume is a smaller fraction of the total breath, so more fresh air gets to the alveoli per minute. Shallow, rapid breathing can actually decrease alveolar ventilation, even if minute ventilation stays the same or increases.
Got it. Okay, let's get into the actual exchange of gases.
In a steady state, the amount of oxygen your body uses must be matched by the oxygen entering your blood and the lungs. And the CO2 produced by your cells must be matched by the CO2 leaving your blood and the lungs.
Makes sense. Balance.
The ratio of CO2 produced to O2 consumed is the respiratory quotient, RQ. It depends on what fuel you're burning. About 1 for pure carbs, 0.7 for fat, 0.8 for protein. On a mixed diet, it's typically around 0.8.
Now, the driving force for gas movement is partial pressure, right?
Exactly. Dalton's law says the total pressure of a gas mixture is the sum of the pressures each gas would exert if it were alone. That individual pressure is the partial pressure, like PO2 for oxygen, PCO2 for carbon dioxide.
And gases diffuse from high partial pressure to low partial pressure.
Always. Atmospheric air is about 21% oxygen. At sea level, 760 mmHg. Atmospheric PO2 is 0.21 by 760 equals 160 mmHg. Atmospheric PCO2 is tiny, almost negligible.
How does this work when gases dissolve in liquids, like blood?
That's governed by Henry's law. The amount of gas that dissolves in a liquid is proportional to its partial pressure in the gas phase above the liquid, and also its solubility in that liquid. Importantly, at equilibrium, the partial pressure of the gas in the liquid is the same as in the gas phase. Diffusion within the liquid also goes from high to low partial pressure.
So what are the typical partial pressures in the alveoli?
Because oxygen is constantly leaving the alveoli to enter the blood, and CO2 is constantly entering from the blood, the alveolar partial pressures are different from atmospheric air. Typical alveolar PO2 is around 105 mmHg, and typical alveolar PCO2 is around 40 mmHg.
What determines these alveolar levels?
Alveolar PO2 depends on atmospheric PO2, how fast you're ventilating your alveoli, and how fast your body is consuming oxygen. Alveolar PCO2 depends on how fast you're ventilating and how fast your body is producing CO2.
So ventilation is key for both.
Absolutely. If your alveolar ventilation doesn't keep up with CO2 production, alveolar PCO2 rises, that's hypoventilation. If your ventilation is excessive relative to CO2 production, alveolar PCO2 falls, that's hyperventilation.
Important note, hyperventilation isn't just breathing fast, it's breathing fast relative to your metabolic rate.
Exactly. During exercise, ventilation increases hugely. But if it matches the increased CO2 production, it's not technically hyperventilation in this physiological sense.
Okay, let's look at the exchange between alveoli and the blood in the pulmonary capillaries.
Systemic venous blood arriving at the lungs is low in oxygen and high in carbon dioxide. Typical values are PO2 around 40 mmHg and PCO2 around 46 mmHg.
And in the alveoli, it's PO2 105 and PCO2 40. Big gradients.
Huge gradients. So oxygen diffuses rapidly from alveoli, 105, into the blood, 40. Carbon dioxide diffuses rapidly from the blood, 46, into the alveoli, 40.
Does the blood fully equilibrate?
In healthy lungs, yes, and very quickly. By the time blood leaves the pulmonary capillaries, its PO2 is typically around 100 mmHg, slightly less than alveolar due to some minor factors, and its PCO2 is 40 mmHg, essentially matching the alveolar air.
What if there's a problem with the lung tissue itself?
If the alveolar membranes thicken, like in diffuse interstitial fibrosis, or if fluid fills the space, like in pulmonary edema, the diffusion distance increases, impairing gas exchange, especially for oxygen. This might only become apparent during exercise when blood flows faster and there's less time for diffusion.
You mentioned VQ matching earlier, ventilation-perfusion matching.
Yes. Ideally, every alveolus should receive an amount of airflow, ventilation V, that's perfectly matched to its blood flow. In reality, there's always some degree of ventilation-perfusion inequality, mostly due to gravity affecting blood flow. This mismatch is actually the most common reason for low arterial oxygen levels, hypoxemia.
So some areas get air but no blood, or blood but no air.
Those are the extremes. Ventilated areas with no blood flow act like extra dead space wasted ventilation. Areas with blood flow but no ventilation act like a shunt, allowing deoxygenated blood to mix with oxygenated blood.
Does mismatch affect CO2 as much as O2?
Generally less so. The body can often compensate better for CO2 imbalances, but mismatching significantly lowers the overall efficiency of oxygen uptake.
Are there local controls to try and fix mismatches?
Yes, quite cleverly. If an area of the lung has low alveolar PO2, meaning poor ventilation, the pulmonary arterioles supplying that area constrict. This diverts blood away from the poorly ventilated region towards better ventilated areas.
And the opposite for airflow.
If an area has poor blood flow, the PCO2 in that area will be low because CO2 isn't being delivered. Low local PCO2 causes local bronchoconstriction, diverting airflow away from the poorly perfused region towards better perfused alveoli. Neat, huh?
Very neat. Okay, blood leaves the lungs oxygenated. What happens at the tissues?
The opposite exchange. Cells are constantly using oxygen, producing CO2. So inside the cells, especially in the mitochondria, PO2 is very low, less than 5 mmHg, and PCO2 is high.
So the gradients are reversed compared to the lungs.
Exactly. Oxygen diffuses from the capillary blood, PO2 100 mmHg, into the interstitial fluid, then into the cells, PO2, 5 millimole Hg. Carbon dioxide diffuses from the cells, high PCO2, into the interstitial fluid, then into the capillary blood, PCO2, 40 millimole Hg.
And as blood flows through the tissue capillaries, its gas levels change.
Right. By the time blood leaves the systemic capillaries and enters the veins, its PO2 has dropped back down to about 40 millimole Hg, and its PCO2 has risen to about 46 millimole Hg, ready for the trip back to the lungs. During exercise, muscle cells use O2 faster, lowering tissue PO2 even more, which deepens the gradient and pulls more O2 off hemoglobin.
Let's talk about that hemoglobin. How is oxygen actually carried?
Only a tiny fraction, maybe 1.5%, is physically dissolved in the plasma water and red blood cell water. At a normal arterial PO2 of 100 millimole Hg, that's only about 3 millimoles of O2 per liter of blood.
That doesn't sound like much.
It's not nearly enough to sustain us. The vast majority, over 98%, is chemically bound to hemoglobin Hb inside the red blood cells. That brings the total O2 content up to about 200 millimolars per liter of blood.
Remind us about hemoglobin's structure.
It's a protein with 4 subunits. Each subunit contains a polypeptide chain, globin, and a non-protein group called heme group. Each heme group has an iron atom, Fe2+, at its center, and that's what actually binds one molecule of O2. So one Hb molecule can bind up to 4 O2 molecules.
We call it deoxyhemoglobin when it's not carrying oxygen and oxyhemoglobin when it is.
Correct. Hb and HbO2. And we measure how much oxygen is bound using percent hemoglobin saturation. That's the amount of O2 actually bound divided by the maximum amount the Hb could bind times 100.
So 100% saturation means all mining sites are full.
Yes. It's important to distinguish saturation from total oxygen content. In anemia, you have less hemoglobin overall. So even if the remaining Hb is 100% saturated, the total oxygen carrying capacity of your blood is reduced.
Let's look at the relationship between PO2 and saturation, the oxygen-hemoglobin dissociation curve.
Right. It's a crucial graph. It plots blood PO2 on the x-axis and percent Hb saturation on the y-axis. And it has a characteristic S-shape, a sigmoid shape.
Why the S-shape?
It's due to cooperativity. When the first oxygen molecule binds to one heme site, it changes the shape of the whole hemoglobin molecule slightly, making it easier for the second, third, and fourth oxygen molecules to bind. Affinity increases as saturation increases.
What are the key features of this curve?
Well, at the top, in the range of PO2 found in pulmonary capillaries, say 70, 100 millHg and above, the curve is quite flat, the plateau. This means Hb is already almost fully saturated, 90, 100%.
What's the significance of the plateau?
It provides a great safety factor. Even if your alveolar PO2 drops moderately, say from 100 down to 60 or 70 mmHg, like at high altitude or with mild lung issues, the Hb saturation only drops a little bit. Oxygen loading in the lungs remains quite effective. It also means that in healthy people, breathing pure oxygen or hyperventilating doesn't add much more oxygen to the blood because the Hb is already nearly full.
Okay, what about the lower part of the curve?
Below about 60 mmHg PO2, the curve becomes much steeper. This is the range of PO2 found in systemic capillaries, especially near active tissues. Here, even a small drop in tissue PO2 causes a large amount of oxygen to be released or unloaded from hemoglobin, perfect for delivery.
So hemoglobin acts like a reservoir.
Absolutely. As dissolved O2 diffuses from blood to tissues, lowering blood PO2, Hb releases its bound O2 to replenish the dissolved pool. This maintains the partial pressure gradient driving diffusion both from alveoli into blood and from blood into tissues. Bound O2 doesn't contribute to PO2, only dissolved O2 does.
Can the curve shift? Can hemoglobin's affinity for oxygen change?
Yes, several factors can shift the curve right or left. A right shift means Hb has a lower affinity for O2. At any given PO2, it unloads O2 more easily. A left shift means higher affinity. It holds onto O2 more tightly.
What causes a right shift?
Increased temperature, increased PCO2, and increased acidity, lower pH, more H+ ions. Think about active tissues. They're warmer, produce more CO2, and are more acidic due to CO2 and lactic acid.
So in active tissues, the curve shifts right.
Exactly. This enhances oxygen unloading precisely where it's needed most. It's a beautiful adaptation. CO2 and H+ actually bind to the globin part of Hb, changing its shape and reducing its O2 affinity.
What about DPG?
Ah, yes. 2-phases dephosphoglycerate, DPG, is a substance produced by red blood cells during their metabolism. DPG also binds to Hb and decreases its oxygen affinity, causing a right shift. DPG levels increase in situations of chronic low oxygen, like high altitude or chronic lung disease, further helping O2 unloading in tissues.
Interesting. What about carbon monoxide again?
Carbon monoxide, CO, is dangerous for 2 reasons. First, as we said, it binds to the O2 binding sites on heme with about 210 times higher affinity than O2. So even low levels of CO can tie up a significant fraction of your HB, reducing oxygen carrying capacity.
Okay, that's bad enough.
But second, CO binding also shifts the O2-HB curve to the left. This means the remaining HB holds onto its oxygen more tightly, making it harder to unload oxygen in the tissues. So it reduces capacity. A&D hinders delivery.
And because arterial PO2 is normal, the body doesn't realize there's a problem.
Exactly. Peripheral chemoreceptors aren't stimulated, ventilation doesn't increase. That's why it's so insidious. Anemia just reduces the amount of HB. CO poisoning reduces the effective amount. A&D poisons the unloading mechanism.
Okay, let's switch to carbon dioxide transport. How does that work?
CO2 is transported in three main forms. About 10% is simply dissolved in the plasma and red blood cell water. CO2 is much more soluble in water than O2 is.
Okay, 10% dissolved. What else?
About 25–30% binds reversibly to amino groups on the globin part of hemoglobin, forming carbaminohemoglobin, HbCO2. Interestingly, deoxyhemoglobin, Hb, binds CO2 more readily than oxyhemoglobin, HbO2.
So unloading O2 in tissues helps load CO2 onto HB.
Yes, it facilitates it, but the largest fraction, about 60–65%, is transported as bicarbonate ions, HCO3.
How is bicarbonate formed?
Inside the red blood cells, CO2 combines with water to form carbonic acid, H2CO3, which then quickly dissociates into a bicarbonate ion, HCO3, and a hydrogen ion, H+.
CO2 plus H2O, but incredibly fast inside red blood cells because they contain the enzyme carbonic anhydrase, Ca.
The enzyme is key. What happens to the bicarbonate then? Most of the HCO3 is then transported out of the red blood cell into the plasma in exchange for a chloride ion, Cl. This swath is called the chloride shift, and it maintains electrical neutrality.
So most CO2 travels in the plasma as bicarbonate, even though it was converted inside the red cell.
Correct. Then in the lungs, as CO2 diffuses out of the blood into the alveoli, all these reactions reverse. Bicarbonate re-enters the red cells, chloride leaves, combines with H+ to form carbonic acid, carbonic anhydrase converts it back to CO2 and water, and the CO2 diffuses out.
What happens to the H+ ions generated during CO2 transport in the tissues? Doesn't that make the blood acidic?
It would, except that most of the H+ gets buffered. And the main buffer here is hemoglobin itself, specifically deoxyhemoglobin.
Deoxyhemoglobin buffers H+ better than oxyhemoglobin.
Much better. As HbO2 releases O2 in the tissues to become Hb, the Hb readily picks up the H+ generated from the CO2 conversion, forming HbH. This minimizes the pH change. Venous blood, pH 7.36, is only slightly more acidic than arterial blood, pH 7.40.
And in the lungs, the reverse happens.
Yes. As Hb binds O2, it releases the H+ cell. This H+ then combines with the HCO3 that came back into the red cell, driving the reaction back towards CO2, which is then exhaled. It's a beautifully integrated system.
But imbalances can occur, leading to pH problems.
Yes. If ventilation is inadequate, hypoventilation, CO2 builds up in the blood, generating excess H+ and causing respiratory acidosis. If ventilation is excessive, hyperventilation, too much CO2 is blown off, reducing H+ and causing respiratory alkalosis.
Okay, who controls all this? How is breathing regulated?
Breathing is controlled by the nervous system. Remember, the diaphragm and intercostal muscles are skeletal muscles. They need signals from motor neurons to contract.
And those signals come from the brainstem.
Primarily the medulla oblongata. There's a region called the medullary respiratory center. Within this, specific groups of neurons fire rhythmically. The dorsal respiratory group, DRG, mainly contains inspiratory neurons that drive the diaphragm and external intercostal.
And the rhythm itself.
The key pacemaker seems to be in the ventral respiratory group, VRG, specifically a network called the pre-Butzinger complex. This generates the basic respiratory rhythm. The VRG also contains expiratory neurons that become active during forced expiration.
Are other brain areas involved?
Yes. The pons, just above the medulla, helps fine-tune the rhythm, smoothing the transitions between inspiration and expiration.
Are there reflexes involved, like from the lungs themselves?
Yes. There are pulmonary stretch receptors in the airway smooth muscle. If the lungs are inflated excessively, these receptors fire and send signals back to the medulla to inhibit inspiration. This is the herringbone reflex, mainly a protective mechanism against overinflation, probably more important during strenuous exercise with large breaths.
And clinically, certain drugs can suppress this medullary center.
Critically so. Opiates, barbiturates, and other sedative hypnotics can severely depress the medullary respiratory neurons, potentially leading to respiratory arrest. That's a major danger of overdose.
How does the body monitor blood gases to adjust breathing?
Through chemoreceptors. There are peripheral chemoreceptors and central chemoreceptors.
Where are the peripheral ones?
Primarily the carotid bodies, located at the branching of the common carotid arteries in your neck, and the aortic bodies on the arch of the aorta. They're strategically placed to monitor the blood going to the brain and the rest of the body.
What do they sense?
They are most strongly stimulated by a significant decrease in arterial PO2, hypoxia, specifically when it drops below about 60 milli-Hg. They also respond to increased arterial H+ concentration, acidosis, and to a lesser extent, increased arterial PCO2. The carotid bodies are the more important ones for respiratory control.
And the central chemoreceptors.
These are located on the surface of the medulla itself. They are extremely sensitive to changes in the H+ concentration of the brain's extracellular fluid.
Not directly blood H+.
Mostly indirectly. While H+ doesn't cross the blood-brain barrier easily, CO2 does. So increased arterial PCO2 leads to increased CO2 in the brain fluid, which then generates H+ locally via carbonic anhydrase, stimulating the central chemoreceptors.
So let's put it together. How does low oxygen affect breathing?
A drop in arterial PO2 below 60 mmHg strongly stimulates the peripheral chemoreceptors, causing a reflex increase in ventilation.
Why the 60 mmHg threshold?
Look back at the O2-HB curve. Above 60 mmHg, you're still on the plateau. HB saturation is still pretty high, around 90%. Oxygen transport isn't severely compromised until PO2 drops below that level, so the reflex kicks in when things start to get critical.
And again, this explains why CO poisoning is tricky. Normal PO2 doesn't trigger this reflex.
Exactly. What about CO2 control?
PCO2 control is much more sensitive. Even a small rise in arterial PCO2 causes a large increase in ventilation.
How does that work?
Increased PCO2 stimulates both peripheral and central chemoreceptors via H+. The central chemoreceptors are actually responsible for about 70% of the response. This powerful reflex keeps arterial PCO2 tightly regulated around 40 mmHg. Can PCO2 get too high?
Yes, and very high levels of CO2 can actually act like an anesthetic and depress ventilation, which is dangerous.
What if blood acidity changes for reasons other than CO2, like lactic acid during exercise?
That's metabolic acidosis. The increased arterial H+ stimulates the peripheral chemoreceptors. Since H+ doesn't easily cross into the brain, this triggers hyperventilation.
How does hyperventilating help?
By blowing off more CO2, you lower arterial PCO2. This, through the chemical reactions, helps reduce the arterial H+ concentration, partially compensating for the metabolic acidosis and bringing pH back towards normal. The reverse happens in metabolic alkalosis, decreased. H+ leads to hypoventilation, raising PCO2 and helping restore pH.
What about ventilation during exercise? It increases massively.
It can increase 20-fold. But interestingly, during moderate exercise, ventilation increases in such perfect proportion to the increased metabolism that arterial PO2, PCO2, and H+ concentration remain remarkably stable.
So the normal chemoreceptor stimuli aren't really changing?
Not significantly, which is a bit of a puzzle. Other factors must be involved. Reflexes from moving joints and muscles, increased body temperature, signals descending from the brain's motor cortex, central command, epinephrine release, maybe even learned responses likely all contribute, especially to the rapid increase at the start of exercise.
And in very strenuous exercise.
Then ventilation often increases even more than CO2 production, causing arterial PCO2 to drop slightly. Lactic acid also builds up, increasing arterial H+Le, which further stimulates ventilation via peripheral chemoreceptors.
Are there other breathing controls?
Sure, protective reflexes like coughing and sneezing, triggered by irritant receptors in the airways. You can voluntarily control breathing to some extent, but involuntary drives, like rising PCO2 when holding your breath, eventually override it. And there are J receptors near lung capillaries, stimulated by fluid buildup, like in heart failure, causing rapid shallow breathing and a sensation of breathlessness or dyspnea.
Okay, let's touch on hypoxia, or oxygen deficiency at the tissue level.
There are generally 4 types. First, hypoxic hypoxia, also called hypoxemia, where arterial PO2 itself is reduced. This is common in lung disease.
What causes hypoxemia?
Things like hypoventilation, not breathing enough, diffusion impairment, thickened alveolar membranes, shunt, blood bypassing ventilated lung, or, most commonly, that ventilation-perfusion inequality we discussed.
The second type.
Anemic hypoxia, or carbon monoxide hypoxia. Here, arterial PO2 is normal, but the total amount of oxygen carried by the blood is reduced because of low hemoglobin or CO interference.
Third.
Ischemic hypoxia, where blood flow to the tissues is inadequate, even if the blood itself is fully oxygenated. Think heart failure or a blocked artery.
And fourth.
Histotoxic hypoxia, where the tissues themselves can't use oxygen properly, even if it's delivered. Cyanide poisoning is a classic example. It blocks cellular respiration.
You mentioned VQ mismatch affects O2 more than CO2. Why?
It relates to the shapes of their transport curves. The O2HB curve plateaus, meaning blood leaving well-ventilated areas, can't carry much extra O2 to compensate for blood from poorly ventilated areas. The CO2 content curve is more linear, so hyperventilation in well-ventilated areas can effectively blow off extra CO2 to compensate for retention in poorly ventilated areas.
One clinical caution.
In some patients with chronic severe lung disease, like COPD, their drive to breathe might rely heavily on the hypoxic stimulus, low PO2, because their response to high PCO2 has become blunted. Giving them high concentrations of oxygen can remove that hypoxic drive and potentially cause them to stop breathing or hypoventilate severely. Needs careful management.
Lastly, the lungs do more than just gas exchange.
Yeah, absolutely.
Yeah.
We've mentioned sound production, phonation. Defense against microbes is huge. They also process certain chemical messengers in the blood, removing some, activating others. And they act as a filter, trapping small blood clots that might form in the legs, preventing them from reaching the brain or heart.
A truly multi-talented organ system.
Absolutely.
So we've really journeyed through the intricate world of respiratory physiology. From those tiny alveoli with their massive surface area, the vital role of surfactant, the physics of airflow governed by Boyle's law, to the complex transport of gases by hemoglobin and the precise neural and chemical controls. It's just an amazing system of balance.
It really is. And we've seen how seemingly small factors, like airway radius or surfactant levels, can have huge impacts, and how the body has multiple backup systems and local controls to try and keep things running smoothly.
And understanding this isn't just academic, is it? It helps explain everyday things like why a deep sigh feels good or why we get breathless at high altitude. And it gives us insight into conditions like asthma, COPD, or even the effects of CO poisoning.
Definitely brings physiology to life.
So here's a thought to leave you with. Consider the incredible complexity we've discussed, all these feedback loops working perfectly to maintain stability despite constant changes inside and out. What might happen, just for a moment, if one of those critical loops failed? What would the cascade effects be?
A sobering thought, highlighting the system's brilliance and fragility. Well, thank you for joining us on this deep dive into the lungs and how we breathe.
From the entire Deep Dive team, thank you for listening.